Hello students, welcome to readmedprep Academy channel. Today we are going to discuss about the microbes in the human welfare part 1. In this we are going to talk about the microbes in household products production, like the production of curd, yogurt, cheese, paneer, dough and the single cell protein. Microbes such as bacteria, fungi, protozoa, certain algae, viruses, viroids and prions are some of the major components of the biological system on Earth. Several microorganisms are beneficial and contribute to the human welfare. Microbes are present everywhere in the soil, water, air and within bodies of animals and plants. Microbes like bacteria and fungi can be grown on nutritive media to form colonies, which can be visibly seen. Some microbes useful to human welfare are discussed here. In this image on the left side, you can see the various types of bacterial colonies. In the image on the right side, you can see the various types of the fungal colonies. In everyday life, microbes and their products are used in the preparation of idli, dosa, cheese, curd, yogurt, dough, bread, vinegar, etc. On the right side, you can see the idli dosa batter. What is this lactic acid bacteria, otherwise shortly called LAB? Bacteria like Lactobacillus acidophilus, Lactobacillus lactis and Streptococcus lactis, commonly called lactic acid bacteria, shortly LAB, are probiotics which check the growth of pathogenic microbes in the stomach and other parts of the digestive tract. In the image on the right side, you can see the lactobacillus. Prebiotics, what are they? They are compounds in food like fibers that induce the growth or activity of beneficial microorganisms. What are probiotics? Probiotics are live microorganisms intended to provide health benefits when consumed, generally by improving or restoring the gut flora. In this image, you can see on the left side the prebiotics, which are foods for the probiotics like the asparagus, garlic, wheat bran, wheat flour, banana. On the right side, you can see the probiotics, which are good bacteria which are beneficial to the gut of the human beings, like yogurt, sour cream, kefir, and probiotic milk. Now, what are the uses of lactic acid bacteria? Lactic acid bacteria is used in medicine, food and feed industries, in the chemical production of many substances and organic acids, so useful in chemistry, and it has a lot of functional ingredients like the probiotics. They are also the starter cultures for the dairy foods and non-dairy foods. They are antimicrobial agents which are used as biopreservatives and in the medicine sector. They have low calorie sweeteners. They are used in the production of exopolysaccharides. They contain vitamins and enzymes production. Now curd. The LAB grows in milk and convert it into curd, thereby digesting the milk protein casein. A small amount of curd added to the fresh milk as a starter or inoculum contains millions of lactobacilli, which under suitable temperature, that is less than or equal to 40 degrees centigrade, multiply and convert the milk into curd. Curd is more nutritious than milk as it contains a number of organic acids and vitamins. Yogurt. Yogurt is produced by the bacterial fermentation of milk, and lactic acid is produced as a byproduct. Microorganisms such as Streptococcus thermophilus and Lactobacillus bulgaricus coagulate the milk protein and convert the lactose in the milk to lactic acid. The flavor in yogurt is due to acetaldehyde. In the images below, you can see the conversion of milk into yogurt. Cheese. Cheese is a dairy product produced in a wide range of flavors, textures, and is formed by coagulation of milk protein casein. During cheese production, milk is usually acidified and the enzyme rennet is added to cause coagulation. The solids are separated. They are pressed to form cheese. Most cheese are made with the starter bacteria of Lactococcus, Lactobacillus or Streptococcus. In this image, you can see the manufacture of cheese. On the right side, raw milk is taken, which is pasteurized, or if not, you can also take unpasteurized milk. Addition of starter culture is done to cause acidification. Addition of rennet is done to cause coagulation of the milk protein, and the way separation is done. Salting of the solids and the shaping of the solid is done by pressing, and the cheese is obtained. Aging is done of the cheese, and it is packed and manufactured and transported and distributed. Paneer. Paneer is otherwise called cottage cheese. It is a fresh cheese common in South Asia, especially in India. It is made by curdling milk with lemon juice, vinegar and other edible acids. Large holes in Swiss cheese is due to the production of large amount of carbon dioxide by the bacterium Propionibacterium shermani. On the right side you can see paneer in a bowl. What is the method of manufacture of paneer? Milk is taken and the standardization of milk is done with fat is to solid not fat in the ratio of 1 is to 1.65, and heat treatment is done at 90 degrees centigrade, and cooling is done at 70 degrees centigrade, and at 70 degrees centigrade 1% citric acid solution is added, which causes coagulation of the milk protein at a pH 5.3 to 5.35, and the whey is separated from the coagulum, and this coagulum is pressed, cut into small blocks. Packaging is done and refrigeration and storage is done and transported and distributed. On the left side above image you can see the production of paneer from milk, where the citric acid is added and the whey is removed and the solid is compressed and cut into small blocks. Dough. The dough used in the preparation of idlis and dosas are fermented by the bacteria Leuconostoc mesenteroids, whereas the dough used in bread making is fermented by Saccharomyces cerevisiae, which is called the baker's yeast. On the right side you can see the dough which is made to ferment. Fermentation of glucose mainly forms ethyl alcohol and carbon dioxide, which is responsible for leavening of dough. When leavened dough is baked, both carbon dioxide and ethyl alcohol evaporate, making the bread porous and soft. In this image you can see on the left side a dough is prepared and is placed in the vessel and is allowed to leaven. After 40 minutes you can see the change where the leavening occurs and increases the size of the dough. Bread must proof to allow the leavening agent to cause the dough to rise before baking. Now single cell protein. Single cell protein refers to edible unicellular microorganisms like spirulina. Protein extracts from pure or mixed cultures of algae, yeast, fungi or bacteria may be used as ingredient or as a substitute for protein-rich foods and is suitable for human consumption or as animal feed. In the image below you can see the sources of the single cell protein like the fungi, the yeast, the algae and the bacteria. So today in Microbes in Human Welfare Part 1, we discussed about how the microbes are used in household products production like the curd, yogurt, cheese, paneer, dough and the single cell protein. So thank you. Kindly subscribe, like, share and comment the channel Read MedPrep Academy. To join Read MedPrep Academy, WhatsApp the number given below. Kindly register for UG and PG NEET type MCQs in our website www.readmedprepacademy.com. Our Facebook ID is Read MedPrep Academy. Our email is readmedprepacademy@gmail.com. Our Instagram is Read MedPrep Academy. Kindly post your questions regarding the lecture in the comment box. We will reply with appropriate answers. Thank you very much.